All right. Hello, everybody. Good morning if you're in Hawaii, or good evening. I know some of you are coming all the way from England, which is wonderful, and elsewhere around the world, Colorado. Thank you all for being here on this really special occasion. It's my pleasure to introduce Rebecca Ward, who's defending her master's thesis today in the Tropical Conservation Biology and Environmental Science master's program here at the University of Hawaii at Hilo. Let me get Bishop in here. My name is Matt Knope. I'm Rebecca's primary thesis advisor, and I just want to give you a little bit of background on Rebecca that kind of leads us up to today. Rebecca already had a really impressive CV before she showed up at the University of Hawaii at Hilo. She graduated magna cum laude, was both a Dean's List and a President's List recipient for her absolutely stellar GPA as an undergraduate student in environmental science at Kennesaw State University in Georgia. And since joining Hilo has really been an integral part of both the TCBES program as well as the greater Hilo Ohana. Just to highlight a few of the many, many things she's done to contribute. She served as the primary instructor for the general biology labs, that's Biology 101. She's also been a teaching assistant for Dr. Pat Hart's ecology labs. She's also a teaching assistant for my advanced tropical island ecology and evolution lab, both of those for 2 years. In addition, she served as the field supervisor for Dr. Hart's research up at Hakalo Forest National Wildlife Refuge. She's now currently employed by the Akaka Foundation, where she's the resource coordinator managing a very large grant that's focused on development of pedagogical resources for K-12, I think actually pre-K-12, right, Becca? Pre-K-12 students throughout the state of Hawaii, building science curriculum, connecting teachers, scientists, and others, doing really incredible work. And having said all of that, you can see just how fortunate we've been to have had Rebecca as an integral part of TCBES, the Tropical Conservation Biology and Environmental Science program, as well as the greater Hilo community. And the future is very bright for her. I'm going to turn it over to her now. I encourage you guys to ask questions, but I'll ask that you hold your questions until the end of the talk. You can either turn your mics on and we'll kind of take them in turn, or you can put them in the chat and I'll try to manage kind of moderating the chat. I'll keep my eye on the chat. After that, I'll ask that once the Q&A is over, that everybody but Rebecca's thesis committee, which is Tracy Wagner, Steve Colbert, and myself, stay on with Rebecca for the closed door defense part of the defense. Okay, having said all of that, Rebecca, the floor is yours.
Thank you so much, Matt. And I just wanted to say thank you so much to everyone that's here from so many different places. It means so much for me to have you here, and me presenting this work would absolutely not be possible without all of you. Okay, so can you guys see my screen? Does everything look good on your end? Awesome. So the title of my talk today is Spatial Variation in Sea Surface Temperature and Antecedent Sea Surface Temperature Conditions Influence Community Structure in the Hawaiian Intertidal. Repeatedly in this talk, I'm going to be referring or using the term antecedent, and if you guys aren't familiar with that, it's another word for prior. Okay, let's get started. This is the outline for the talk today. I'll start off giving you guys some background on global and local trends in sea surface temperature and introduce you to Hawaii's intertidal. I'll go through the objective of my study, which is to look at how are Hawaiian intertidal communities responding to sea surface temperature change. I'll go through my methods, characterizing sea surface temperature, looking at relationships across multiple spatial scales. I'll go through my results, looking at warming trends, how antecedent sea surface temperatures are standing out as a major influencer of change in Hawaii's intertidal, and then go through discussions. Another thing to point out is that throughout the course of this presentation, my citations are numbered rather than listed out. So if you're curious about a source, you can write down the reference number I also, in the chat, which I don't know if you guys can see it, or maybe Matt can copy it and send it again. There's a link to the PDF of the list of resources if you guys would like to reference them while I'm presenting. Okay. So let's get started with some background. The warming of Earth's oceans and its relationship to human-driven climate change is now a well-documented phenomenon. This graphic is a really great visualization of global anomalies in temperature. I didn't make the graphic, but I think it so clearly illustrates that yes, warming is indeed happening and the rate of warming is increasing exponentially. And we see this within Hawaii's intertidal and within Hawaii as a whole. Here, warming trends are expected to increase from the historical rate of 0.16 degrees Celsius per decade to 0.39 degrees Celsius per decade by the end of the century. And this is an almost 250% increase in the rate of warming. Now, as researchers have documented these rising temperatures, both within Hawaii and across the globe, we've observed countless diverse consequences within marine systems. And these consequences might advantage or disadvantage certain taxa through a number of mechanisms. Here I've listed only a few processes within marine communities that are linked to sea surface temperature, and this isn't exhaustive at all. So things like the metabolic use of oxygen, larval dispersal and settlement, predation rates, sea surface temperature can influence sex ratios for species where sex isn't determined at fertilization. For algal communities, it's been linked to shifts in growth and overall biomass. And so there's this intimate connection between ocean temperature and the physiological processes of marine life. So sea surface temperature, it stands out as a driver or predictor of flux in marine systems. And it really is a climatic variable that warrants close monitoring and study. These interactions between sea surface temperature and marine organisms aren't just seen in the open ocean. The intertidal is among the many ecosystems that are vulnerable to the influences of sea surface temperature change. Just as a quick refresher, intertidal is that land between the highest high tide and the lowest low tide. The organisms that live in the intertidal have to cope with really rapidly changing environmental factors, things like drying out, shifts in salinity, daily temperature changes. So they're resilient, but even though they're resilient, they're still vulnerable to changes in environmental conditions, and they display responses such as the ones that I listed on the previous slide. Interestingly, intertidal ecological communities can be very different from one another, even across small distances. So two intertidal sites just a few miles apart might have drastically different community structures within them. And continued research is revealing that it's the effect of biotic and abiotic variables that help shape these systems. So differences in substrate type, influx of nutrients, grazing, predation, competition, disturbance by humans or human development. All of this can contribute to differences in community structure between intertidal sites. So the intertidal is this complex habitat. And this complexity is also apparent when observing the responses to changing sea surface temperatures. Globally, intertidal communities appear to be responding to warming temperatures across multiple temporal scales or time scales. So short-term trends in sea surface temperature like annual seasonality have been identified as significant drivers for shifts in algal biomass across a year. Long-term trends like multiple years to decades-long trends in sea surface temperature warming have been found to strongly influence intertidal communities, leading to shifts in the range or the abundance of entire populations in as little as a decade. And also several studies have found that biological responses to change in temperature can be delayed and may not even be apparent until weeks or months later. And this is a study that found that standing algal biomass was most sensitive to sea surface temperature conditions 3 to 4 weeks prior. So with the significance of both short and long-term trends in sea surface temperature and the potential for delayed responses, there's a need to account for multiple temporal scales when investigating the effects of sea surface temperature within intertidal systems. So while temperate intertidal systems in places like California, Oregon, and Washington have a long history of intensive scientific study, intertidal systems in more geographically isolated locations, such as Hawaii, have only begun to be investigated. So although Hawaii has a pretty small tidal range, about 1 meter between high and low tides, it's home to a diverse assemblage of fishes, algae, and invertebrates. And it has a greater presence of endemic and non-native species than what's seen in many temperate regions. The harvest of algae and invertebrates and fishes carries strong cultural and economic significance here in Hawaii. And just like other intertidal sites across the world have been found to be very variable, we've also found that in Hawaii. So Hawaiian intertidal communities, both between sites and across islands, are very different from one another, and site level differences in substrate, topography, and wind and wave exposure have all been suggested as factors that influence local community composition. Notably, one study in Ewa Beach on the southwest side of Oahu linked yearly variation in algal composition to maximum water temperature. This study was only conducted at Ewa Beach, so just one site, but it serves as a critical first step to understanding, what kind of relationship is going on between ocean warming and Hawaii's intertidal communities. And if sea surface temperatures in Hawaii do warm according to the projected trends, we need to understand both how individual taxa and entire communities are going to respond. So this leads us to the guiding question for my study, which is how are Hawaiian intertidal taxa and communities responding to sea surface temperature change? And there are a few considerations to take into account when seeking to answer this guiding question. As we've explored, intertidal systems around the world and in Hawaii are very variable. And because of this large variability, analyses that are limited to just a single intertidal site may not be representative of trends across all of Hawaii. So to effectively capture these larger responses to change, there's a need for investigations to be conducted across multiple spatial scales. Further, as previous studies outside of Hawaii have shown intertidal communities to have both immediate and delayed responses to short and long-term shifts in water temperature, I want my analyses to be conducted in a way that makes it possible to identify windows of time when sea surface temperature is the most influential. For this reason, it's important for me to incorporate multiple timescales and the ability to pinpoint which time windows, if any, are significant. And finally, I want analyses looking at changes in sea surface temperature to be conducted with the highest resolution data possible. I love this GIF. It was created by the Naval Research Laboratory, and I think it's a really wonderful visualization of sea surface temperature trends in Hawaii. This is just over 2019. And you can see that temperature conditions can vary just a lot from one side of an island to the other. So if these differences in sea surface temperature trends are significant, I want to be able to pick up on that and use it to inform my modeling. So to accomplish this research objective, my study addresses the following 3 research questions. And I'll just break them down one at a time. The first is, has sea surface temperature changed across the main Hawaiian Islands, both for the main Hawaiian Islands as a whole and across different sites over the course of the study's timeframe? So before asking, how have intertidal communities and taxa changed in response to sea surface temperature, there's a need to understand, has sea surface temperature changed in a meaningful way, and also to define that over the course of my study. Second, at the site level, are individual taxa are individual algal and invertebrate taxa and the diversity of these intertidal ecological communities responding to changes in sea surface temperature. And then finally, across the main Hawaiian Islands, are individual algal or invertebrate taxa and/or the diversity of these intertidal communities responding to changes in sea surface temperature. And again, intertidal sites in Hawaii and across the world are known to be very different from one another. So this question attempts to address, even with that high variability between sites, are there still trends that can be observed across the state as a whole? This brings us to the methods. So this image is of my 12 sites for this study. There are 12 intertidal sites. The majority are on Oahu, 3 are on Hawaii Island, 2 on Molokai, and one is on Maui. At each site, intertidal algal and invertebrate community composition data was collected by the Educational Citizen Science program called Our Project in Hawaii's Intertidal, or OPIHI for short. Since 2005, OPIHI has paired professional marine scientists with secondary school students and teachers to collect algal and invertebrate community level data all across the state, which is amazing. So since 2004, OPIHI has sampled 46 intertidal sites across the Hawaiian archipelago. And there is an 8-year gap in data collection that happened from 2008 to 2015, and that was due to a lack of funding for the program. So the 12 sites that were selected for this study are those with data collected both before and after that 8-year gap in data collection. And that was just so that I could best assess what's happening over time in Hawaii. It's important to point out that OPIHI data has been found to be of comparable quality to data collected by experts in the field. It's high quality, but also that data is most appropriate for monitoring more abundant, morphologically distinct species rather than those that are rare or cryptic. So how this data is collected, at each site, 3 to 7 transects were laid perpendicular to shore, similar to what you see in the photograph here on the left. And they were laid at different varying lengths and distances apart from one another, depending on the location, topography, and also safety for students. A quadrat point intercept method was used to survey. So gridded 0.3 by 0.3 quadrats with 25 point intercepts were set along the transect lines at regular intervals. And within each quadrat, any organism directly below one of those 25 point intercepts was identified to the lowest taxonomic level or functional group possible. And although all the organisms in this study were identified at different levels, I'm using the generic term taxa to refer to all of the organisms here in the study. And all of this data is made publicly available through the OPIHI's website. So once that data was downloaded from the OPIHI website, I took it through a round of post-processing. Cryptic or rare species were grouped at higher taxonomic levels. The 3 algae taxa that you see at the bottom of the screen are really great examples of this. So there are so many species of the algae Podyna in Hawaii that are often very difficult to differentiate between. So this is one that was grouped, any Podyna that were identified by students was grouped at the genus level. Similarly, there are several genus of brown crustose algae in Hawaii that are very similar in appearance. So again, all of them were grouped and referred to as a single taxa. And also turf algae, which as some of you may know, it's a conglomerate of many different species of microalgae. It's referred to here as a single taxon for simplicity. On the other hand, morphologically distinct or dominant species were retained at the species level. Again, 2 examples, Turbinaria ornata, which is an abundant native species here in Hawaii, and the invasive species Acanthophora spicifera. They're both morphologically distinct and pretty easy to ID, so they were retained at the species level. So following biological data processing and filtering, a total of 116 algal and invertebrate taxa were identified across all 12 intertidal sites. The most abundant taxa were identified for each of the 12 sites and for the main Hawaiian Islands as a whole. To capture changes in the overall community, Simpson's diversity index value was then calculated for each sampling day. Although the earliest OPIHI data in the study was collected in 2005, I obtained sea surface temperature data from 2004 up through 2019. And this was to provide insight into antecedent climatic conditions up to a year prior to each sampling date. Sea surface temperature data was produced by OISTA, which is the operational sea surface temperature and sea ice analysis product. This is compiled by Copernicus, which is run by EU's Earth Observation Program. The OISTA data are collected both by infrared and microwave satellite measurements, in addition to in situ data from both moored and drifting buoys. And they use all this information to calculate daily sea surface temperature at a resolution of 1/20th of a degree, which in Hawaii is approximately 6 kilometers squared. So to detect variation in sea surface temperature trends, as near as possible to each of my intertidal sites, the daily sea surface temperature was extracted from that 6 kilometer squared pixel encompassing each intertidal location. Both near and reprocessed oyster products were used to obtain daily sea surface temperature measurements. And these are just two different products. They come from the same data source, but it's different ways of analyzing the satellite data. Before using both products, I ran a series of correlation tests and prediction intervals to verify that the two sources were highly correlated with little variability between them. And then finally, to detect any trends in sea surface temperature across the main Hawaiian Islands, I generated average daily sea surface temperature for the total area encompassing the state, which was about 279,000 kilometers squared. Okay, so now we're getting into the statistics. I put that really beautiful GIF back up because I think it's amazing. So starting off, I wanted to understand the linear rate of change from 2004 to 2019. So this was calculated for the main Hawaiian Islands as a whole, and then also for each of the 12 intertidal sites. And all of them were plotted to compare differences in warming trends. Average daily sea surface temperature measurements for the archipelago were then decomposed into seasonal, trend, and irregular components using moving averages. Decomposing the sea surface temperature time series allows me to visualize any trends in the data once seasonality has been accounted for. To identify and understand the effect of any highly influential data points on warming trends, so this is things like outlying extreme summer or winter temperatures, I used Cook's distance to calculate, distance to pick out any of those highly influential data points and to see the effect of these influential data points on the warming trend observed. And then finally, if you remember, OPIHI experienced that gap in funding and there was no data collected from 2008 to 2015. So because of this 8-year gap occurred right in the middle of my studies time frame, I wanted to understand how sea surface varied between the two sampling periods. So a series of two-tailed t-tests were used to determine if the mean, max, minimum, and variance of yearly sea surface temperature across the main Hawaiian Islands differed significantly from the first and second half of the study's time frame. The R package ClimWin was used to model the relationship between sea surface temperature and the study's biological responses, which in this case is point count abundance and diversity. To better account for the influence of sea surface temperature in its modeling, ClimWin detects the time windows preceding ecological sampling over which the biological responses are the most sensitive to sea surface temperature. Daily sea surface temperature data were collapsed into monthly statistics. So where every month, the significance of the minimum, maximum, and average sea surface temperature were compared across alternative time windows. And the goodness of fit among alternative time windows was assessed by AIC values. Each model's optimal time window was reported as the range of months prior to ecological sampling in which the trends in sea surface temperature were the most influential for the biological response. And two groups of analyses were conducted. The first group consisted of site-level examinations, and the second group looked at the main Hawaiian Islands as a whole with the data from all sites compiled and assessed together. So as I mentioned earlier, OPIHI data best documents for the more dominant taxa and intertidal communities. So for this reason, abundance analyses for the study were limited to the most abundant taxa. At each intertidal location, the 5 most abundant taxa were ID'd based on average daily point count abundance. And the relationship between sea surface temperature and point count abundance was then independently assessed for each of the 5 taxa in ClimWin using a negative binomial regression. And a sample of that, what that model looks like, is in italics below. And a negative binomial regression was chosen for these analyses as it best accounted for the strong zero inflation and over dispersion seen in the data. and it still remained compatible with the ClimWin program. So a total of 60 negative binomial models were run, one for each of the 5 most abundant taxa at each of the 12 intertidal sites. Linear regressions were then run in ClimWin to assess the relationship between sea surface temperature and Simpson's diversity at each site. Again, a sample of that model is in italics. A total of 12 linear regressions were run, one for each of the 12 intertidal sites. And finally, a generalized dissimilarity model, or GDM, was used to identify significant drivers of variability between the 12 intertidal sites. So not only do GDMs identify potentially influential predictor variables, but they also provide the rate of compositional change that occurs across the gradient of the environmental predictor variables. So variation across all 12 intertidal sites was modeled as a function of 5 possible influencing variables. So there was distance, rate of sea surface temperature warming, the mean sea surface temperature from the first half of the study's time frame, mean sea surface temperature from the second half of the study's time frame, and then mean sea surface temperature across all sampling years. And variables that significantly influence the data were identified, and the percent deviance explained why the model was generated. So for the main Hawaiian Island analyses, among all intertidal sites cumulatively, the 10 most abundant taxa were identified based on average daily point count abundance. And I looked at the 10 most dominant taxa instead of 5 in order to more appropriately capture the range of taxa with potentially key ecological roles. This expansion also accounted for any site level differences in community composition, as many taxa were only found as dominant community members in one site. And the relationship between sea surface temperature and the point count abundance of each taxon was then independently assessed in ClimWin, again, with a negative binomial regression. Linear regression was also used to assess how Simpson's diversity index had cumulatively changed across all 12 intertidal sites. And to account for differences in sea surface temperature between intertidal locations, the biological data from each site was paired with the sea surface temperature measurements for that location, rather than just using an averaged, statewide sea surface temperature value for all sites. And this was again used to increase my ability to pick up on any fine scale impacts of sea surface temperature. Okay, let's go to results. And I'll start off with the temperature analyses results. So if there's anything that you need to take away from this talk, it's that over the course of the study, Hawaii's ocean waters warmed, and they warmed at a rapid rate. So the results from the linear regression indicated a rate of warming for the main Hawaiian Islands that of about 0.4 degrees Celsius per decade. And this is slightly greater than the projected rate of warming for the end of the 21st century. Outlying data points were not found to be driving this warming trend. So confidence is high that the changes in temperature experienced over the course of the study are true warming trends and not driven by anomalous sea surface temperature events. When I compared temperature conditions from the 2 halves of the study's time frame, maximum sea surface temperature in the second half of the study's time frame were marginally significantly greater. And this further affirms that there was a change in temperature conditions and that warming occurred over the course of this study. The box plots that you see here are just a summary of the data year by year. The middle dark line in each box plot is the median, and I drew those red boxes around each time period to help you visually see that the maximum temperatures over the latter half of the study were higher. All 12 sites experienced a similar high rate of warming. So the linear regressions of daily sea surface temperature across all 12 sites have revealed warming trends between 0.34 and 0.41 degrees Celsius per decade. And although the rates of warming were similar, all falling within 0.07 degrees Celsius of one another, the sea surface temperature conditions at each location appeared to differ. So for example, sea surface temperature conditions at those top 3 sites in yellow, orange, and red, Mealy Point, Barbers Point, and Eva Beach, appeared to be warmer than compared to the rest of the intertidal locations. All of these sites, regardless of where they started off, experienced the same high rate of warming, similar to what was predicted for 80 years from now. So these analyses give us a really great idea of how sea surface temperature conditions have changed over the course of the study, that warming occurred at a high rate with greater max sea surface temperatures in the latter half of the study, and that while the rate of warming is similar across the main Hawaiian Islands and all sites, there's still differences in sea surface temperature conditions between locations. And now that we have a grasp on how sea surface temperature conditions have changed over the study, we're going to look at now the effect of that change on the biological responses, starting off with the site level results. So as I mentioned previously, many studies have demonstrated that Hawaiian intertidal communities vary greatly from one another. And yes, my site level analyses really supported those previous work. I found high variability in community and taxa responses to sea surface temperature among all intertidal sites. The 5 most abundant taxa often differed between intertidal sites, but taxa that occurred the most frequently as one of those top 5 were taxa like turf algae, Padina, Acanthophora spicifera, which is an invasive, and brown crustose algae. And results from the Klimwin analyses revealed that there was a lot of variability in how these taxon were responding to sea surface temperature. In particular, the strength and direction of that relationship was variable. Whatever the influential sea surface temperature variable also differed, and also the most significant time window all differed from site to site. Simpson's diversity was significantly related to sea surface temperature at 8 out of 12 sites, but again, the strength and direction of the relationship was also found to be variable from site to site. So the GDM of community compositional dissimilarity across all 12 intertidal sites, it tested for the significance of those 5 predictor variables. And the rate of sea surface temperature warming and mean sea surface temperature from the second half of the study's timeframe were not found to be significant. So they were excluded from the full model. The results of the model containing the 3 significant variables, which is mean sea surface temperature from the first half of the timeframe, mean sea surface temperature across all sampling years, and geographic distance, that's displayed here. So for each plot, the curve's maximum height represents the total amount of community dissimilarity associated with the variable. The slope of the line represents the rate at which community dissimilarity increases along the variable's gradient. So the results of this model explain 45% of deviance in community composition between sites. The model results provide magnitude for each predictor variable, which is a measure of how influential each variable is for explaining community dissimilarity. So mean sea surface temperature from the first half of the study's timeframe was found to be the most influential variable for explaining dissimilarity between intertidal communities, followed then by mean sea surface temperature across all sampling years, and to a much smaller degree, but still significant, geographic distance. So I found the intertidal sites to be highly variable from one another in regard to the responses to sea surface temperature. And these sites aren't just different from one another because they're separated by distance. Temperature, and in particular, sea surface temperatures from the several years prior, significantly explained 45% of the differences in community composition between intertidal sites. So this brings us to the set of analyses for the main Hawaiian Islands. And even though my intertidal sites were so variable from one another, trends were detected for the main Hawaiian Islands as a whole. So to recap, the relationship between sea surface temperature and abundance was assessed for the 10 most abundant taxa across all 12 sites within the main Hawaiian Islands. And only 6 of the 10 taxon assessed were found to have a significant relationship with sea surface temperature. And I'm going to go through each of those 6 one at a time. So on the right, there's going to be a table that's filling in. It'll have the taxon's name, the mean percent abundance, and the relationship between sea surface temperature and the abundance of that taxa. And then on the left is the negative binomial plot. And this is the modeled relationship. The modeled relationship itself is indicated by the black line, and then the 95% confidence interval is in dark gray. Climate statistics and time windows found to be influential are listed on the x-axis, and the y-axis is percent abundance. So right now, all I have up is turf algae. Turf algae was the most dominant taxa by far across the main Hawaiian Islands, and it was found to have a negative relationship with sea surface temperature warming. So just to further clarify how to interpret this, warming mean sea surface temperature conditions, the 3 to 11 months prior, were found to have a negative effect on the abundance of turf algae, with a 10% decline in abundance for every 0.1 degrees Celsius increase in sea surface temperature. The next 2 most abundant taxa with significant relationships were Padina and Acanthophora spicifera, both with positive relationships between their abundance and sea surface temperature. A negative relationship was found for brown crustose algae and for the native taxa Turbinaria ornata. And finally, Dendropoma gregarium, which is an invertebrate also known as a gregarious worm snail, was found to have a positive relationship. For 5 of these dominant taxa, with the exception of brown crustose algae, the time windows most influential for describing the observed trends in abundance was 6 months wide or wider. In particular, for these taxa, those wider time windows ranging the 1 to 12 months preceding ecological sampling were the most influential for describing changes in abundance across the main Hawaiian Islands. These results are again highlighting antecedent sea surface temperatures over the many months prior as significant drivers of change within Hawaiian intertidal communities. And it further complements the findings from the GDM that antecedent sea surface temperature conditions are influencing community dissimilarity between intertidal sites. An important thing to note is that the y-axes of these plots are all scaled differently just to allow you to see the relationship between sea surface temperature in each of the taxa. And this is because of how abundant each taxa tends to be across Hawaii varies. And you can see that in the table on the right. So turf algae has an average abundance of about 22%, and a taxa like Dendropoma gregarium has an average abundance very near to 1%. So how a change in abundance across these taxa affects overall community composition is going to look different. For example, a 10% decrease in turf algae from its average percent abundance of 20% would look like a 2% decrease in overall abundance. But for a taxa like Dendropoma gregarium, a 10% decrease from its average percent abundance of 1% would look like a 0.1% decrease in abundance. So if I were to set the scales of all these plots from 0 to 100, it becomes much harder to visualize those significant relationships for the taxa with lower abundance. At the same time, with everything on the same scale, it's easier to see that an organism like turf algae might have a more visible impact on changes to overall community composition as they respond to changes in sea surface temperature. And the realities of this are seen in the results of the diversity analysis. When Simpson's diversity index was analyzed across the main Hawaiian Islands, a significant 1.5% increase in diversity was identified for every 0.1 degrees Celsius increase in sea surface temperature. Similar to the highly abundant taxa, trends in sea surface temperature over a wide time window, the 1 to 10 months preceding sampling, were the most influential for describing the observed increase in diversity. This positive shift in Simpson's diversity index was found to be driven by a decrease in the abundance of turf algae across all intertidal sites. So Simpson's diversity is a measure of both the number of species in a community and their relative abundance. And when you pull down the abundance of the most dominant taxa, in this case turf algae, as it's coming down, other species have the ability to become more prominent, to become more apparent more evenly. To illustrate this further, turf algae was removed from the diversity analyses, and a negative, insignificant relationship was revealed between sea surface temperature and significant relationship was revealed between sea surface temperature and Simpson's diversity, just further illustrating turf algae's fundamental effect on community composition at these intertidal sites. So in summary of the main Hawaiian Island results, across the main Hawaiian Islands, a significant relationship was found between sea surface temperature, dominant taxa, and diversity. And for 5 out of 6 of these dominant taxa, and for diversity also, trends and antecedent sea surface temperature conditions over the several months preceding sampling best explained changes in abundance. Okay, so this brings us to the discussion. And I think it's important to start off looking at the rate of sea surface temperature warming that was found over the time frame of the study. So the observed rate of warming, both for the main Hawaiian Islands as a whole and across the 12 sites, fell within the high end of warming projections for Hawaii, and actually just barely surpassed predicted rates of warming for the end of the 21st century. In addition, the second half of the significantly greater maximum yearly temperatures, and those are like the summer highs. So again, these findings align with projections of how sea surface temperatures are going to change in Hawaii, that summers are going to be getting warmer, that the rates of warming will continue to increase. But it's really alarming that we're observing these trends now. Even further, it's significant that this warming was found to be influential for those key dominant taxa and overall community composition, both at the site level and across the main Hawaiian Islands. When we look at the results for each of the sites independently, there was a lot of variability in these responses to sea surface temperature warming. Previous studies have demonstrated that Hawaiian intertidal communities can vary greatly from one another, with interactions between local biotic and abiotic factors driving differences in community composition that are often difficult to predict. My site-level analyses support these findings, and they indicate high variability in community responses to sea surface temperature among intertidal sites. The results of my GDM build on the work done by Cox et al. In 2017 at Ewa Beach, and again highlight the significance of water temperature for determining community composition in Hawaii's intertidal. Even though the observed warming trends were similar across the main Hawaiian Islands and among the 12 sites, variability in sea surface temperature conditions across sites was significant for describing differences in community composition. Additionally, the results from my GDM and the main Hawaiian Island analyses bring to light the significance of antecedent sea surface temperatures within these systems. These results point to the lasting influence of local antecedent sea surface temperatures on shaping intertidal community composition, and suggest that these slight differences are sufficient to affect community structure. It's possible that continued shifts in community composition will be observed in the coming years as rates of warming and baseline temperatures are projected to increase. The set of main Hawaiian Island analyses really highlighted how sea surface temperature is influencing intertidal communities at a larger spatial scale. Studies within many temperate intertidal regions of the world have also found shifts in sea surface temperature to strongly influence community composition at large spatial scales. In particular, a landmark study by Berry et al. In the 1990s found that warming sea surface temperatures over the course of a decade resulted in significant shifts to the range and relative abundance of entire populations in Monterey Bay in California. And multiple factors that co-vary with sea surface temperature may be driving this. And I listed a lot of those variables in the beginning of this presentation. Again, subtle differences in sea surface temperature are known to influence larval success, dispersal, predation rates, sex ratios, and growth rates. And all of this can tie in the effect of sea surface temperature to the biological communities. And we found 6 highly abundant taxa within Hawaii to be responding. And these taxa play key ecological roles in Hawaii. So they are primary producers, they're carbon sinks for the taxa that are known to sequester carbon dioxide from the ocean. They serve as erosion control. Dendropomo gregarium can form huge benches that act almost as like wave blocks, keeping waves from eroding away the shoreline. And also some of the taxa are invasive threats like Acanthophora spicifera. So changes to these key community members may result in shifts in community dynamics and food web dynamics in years to come. And further study is needed to understand what kind of shifts we can expect. So for the majority of those abundant taxa, sea surface temperature conditions, the 1 to 12 months prior significantly influenced their abundance. Some previous studies have identified narrower time windows, so like the 3 to 4 weeks prior or annual seasonality, as influential to algae and intertidal and reef habitats. While the trends associated with these narrow time windows describe interannual fluctuations in the intertidal, they may be too limited to define how these communities are responding to decadal changes in temperature. By determining the significance of long-term warming sea surface temperature trends over the several months preceding ecological sampling, my study further highlights the need for wider sampling timeframes in intertidal communities. If Hawaiian intertidal communities' structure is shaped by antecedent sea surface temperature conditions, it's possible that continued shifts in community composition will be observed in the coming years. So for future implications, sea surface temperature warming trends are known to be localized or dampened due to the influences of groundwater influx, and nutrient influx has long been connected to algal blooms. Future research could build on my work by incorporating these additional abiotic factors, among others. With subtle differences in antecedent sea surface temperature between sites being found as significant and as a significant influencer of communities, I'd recommend that future studies within the Hawaiian intertidal be conducted at the finest spatial and temporal resolutions possible. So satellite sources such as the oyster products I used in this study are an excellent resource for obtaining longer-term climatic data at a relatively fine spatial scale when in situ monitoring is not a possibility. Further, continued funding and support for citizen science initiatives such as OPIHI is so important and could provide valuable long-term, high-quality sources of data for intertidal ecological communities all across the state. Responses to sea surface temperature at each of the 12 sites could be further explored on their own. Additionally, the modeled results of this study could be teased apart further to explore site-level trends in sea surface temperature diversity and abundance with additional abiotic and biotic variables. Okay, in conclusion, this study offers a first look into intertidal community responses to elevated rates of warming. Ultimately, the sea surface temperature trends observed over the time frame of the study capture a period of rapid warming for the Hawaiian intertidal, similar to what's been projected for the end of the 21st century. Differences in antecedent sea surface temperature conditions significantly described overall community dissimilarity between sites and across the main Hawaiian Islands. And antecedent sea surface temperatures over the several months preceding ecological sampling were identified as significant drivers of change within Hawaiian intertidal communities. These results highlight that although Hawaiian intertidal algal and invertebrate communities are highly variable among sites and islands, antecedent sea surface temperatures have a lasting influence on shaping intertidal communities across the main Hawaiian Islands. And with that, I want to say a big mahalo to everyone that has made this work possible. Thank you to my committee, Matt, for your many hours of guidance, Dr. Colbert for your help with oceanography and guidance in my climate analyses. Tracy, thank you for supporting me at the finish line, giving me the support I needed to reach the end. I want to thank all of my collaborators, all of the amazing people at Opihi, Flo, Joe, Jessica. Thank you to Ian Ware for his guidance on all of the stats, my assistant researchers who we all spent hours poring over the data and grouping taxa. Thank you to my TCBES community, all the professors in my cohort, Pat Hart, Becky Ostertag. Thank you to my family for their support. And then also I just want to say a huge mahalo to my community just for lifting me up over the past 1.5 years that's been so challenging. All the love and laughter, surf sessions, long runs, good food truly made it possible for me to keep going even when I was feeling depleted. So I'll always be so grateful to all of you. Aloha nui loa and mahalo from the bottom of my heart. Thank you.